Hi, I'm JP, and I'm going to explain how early onset Alzheimer's disease develops within the human brain, while also explaining the physiological methods on every step within the process. Early onset Alzheimer's disease is very different from late onset Alzheimer's disease. Early onset affects younger people before the age of 65. This affects 5% of the Alzheimer's population within the United States. Especially, out of the 4 million people who are affected from Alzheimer's disease, 200,000 of those are affected by early onset. This is specifically meant from the genes that are expressed within the human genome, completely different with late onset. Early onset Alzheimer's disease is one of the most prevalent forms of dementia. It specifically causes changes in memory, thinking, and behavior. But the difference between the symptoms in early onset and late onset is that there are more systematic physiological changes. In early onset Alzheimer's disease, we see a specific change within the vision. Vision is changed to atrophy within the posterior cortical. After that, we usually see changes in speech, also known as logopenic aphasia. And then finally, one of the hallmark traits is a lack of decision and planning skills within their behavior. Seeing these three are a red flag for diagnosing early onset Alzheimer's disease. Although Alzheimer's disease in general is a very crippling disease, these three can allow Alzheimer's to become more prevalent at the beginning than when it should be. Early onset Alzheimer's is a type of disease that is called familiar Alzheimer's disease. It specifically runs linked to three type of genes: APP, PSEN1, and PSEN2, which is different from late onset Alzheimer's disease, which is APOE. And the thing is that the genetics behind Alzheimer's is that Alzheimer's in general is a single gene mutation disorder. However, there are a multitude of genes which can mutate to cause one of the many variants of the disease. They increase the person's genetic risk by getting more gene variants. Early onset Alzheimer's is inherited through a dominant autosomal method. If one parent has the disease, there's a 50-50 chance that their children will also have it. Mutations in the chromosome create increased amounts of beta amyloid proteins, which cause a major component of plaques. And mutations also create abnormal amyloid precursor proteins, APP, which cause plaque formation also. This is the reason that early onset Alzheimer's disease is called a familial Alzheimer's disease, due to its passing down of genetic mutations. In Alzheimer's disease, there's an overall shrinkage in brain tissue. The grooves or furrows in the brain called the sulci are noticeably widened, and there's a shrinkage within the gyri. In addition, the ventricles or chambers where the brain's cerebrospinal fluid is held, we see are enlarged. Specifically, when we see short-term memory losses, that's when the hippocampus begins to degenerate. Amyloid plaques are made up of beta amyloids, also known as a protein fragment, snipped from an amyloid precursor protein. In a healthy brain, these protein fragments are broken down. In Alzheimer's disease, the fragments accumulate to form hard, insoluble plaques. Neurofibrillary tangles are insoluble twisted fibers found inside the brain cells. These tangles consist primarily of proteins called tau. They help produce the microtubules. In Alzheimer's disease, however, the tau protein is abnormal and the microtubule structure collapses, causing degeneration. Beta amyloid is derived from the amyloid precursor protein APP. It's subsequently cleaved by the enzymes beta and gamma. The precise physiological functions of APP and beta amyloid is not well understood. In Alzheimer's brains, it's not degraded properly, resulting in the formation of toxic oligomers and aggregates of amyloid fibrils, which we can see in the left. These fibrils exist, causing therefore neural degeneration and a lack of communication within the neuron. Tau filaments are the main component of neurofibrillary tangles, composed within the Alzheimer's markers. As a microtubule-associated protein, tau normally binds and stabilizes the microtubules. But in Alzheimer's, it becomes hyperphosphorylated, causing misfolds and eventually aggregates, forming the tangles. It leads to the breakdown of the neuron's transport system, and due to the breakdown, it causes the degeneration within the neuron. The amyloid cascade hypothesis explains how beta amyloid plaques are formed and how they're triggering within Alzheimer's disease. The gradual changes cause a change within the brain cascade of pathological events. Changes in amyloid beta causes a particular change in missense mutations within the specific genes that cause Alzheimer's disease, also known as APP, PSEN1, and PSEN2. The relative changes within the beta amyloid causes an increase of oligomer formation, which causes subtle and increasingly severe synaptic functions. Through through that, the plaques and local inflammatory glial cells affect the microglia and the astrocytes. The synaptic spine loss and neurotic dystrophy occurs. Over time, these changes cause oxidative stress and altered ionic neuronal homeostasis. And without homeostasis, we don't have a balanced home to have a form of healthy neuron. The protein tau is then hypophosphorylated, like explained beforehand. And then aberrant oligomerization is occurring. The development of neurofibrillary tangles are then happening. The cascade accumulates with a widespread synaptic and neuronal dysfunction, and the cell eventually dies. The cascade is so progressive that dementia begins to occur, and the extensive beta amyloid and tau pathology causes such, which will then repeat and reoccur until more severity, causing a change in anatomical differences within the brain. Throughout this video, we have been explaining how Alzheimer's disease within early onset affects the brain, but we must also understand not just what the amyloid plaques and what the tau proteins do, but what steps it causes for the anatomical changes within an Alzheimer's brain. As we can tell from the pictures before, there's a structural change within the hippocampus, within the ventricles, and the cortex. And right now I will explain what steps it takes to get there. There are plenty of formations. The beta amyloid deposition in the brain causes spatial progression starting at the basal neocortex. It spreads throughout the hippocampus and it eventually spreads to the rest of the cortex. The spread of the tau pathology is completely different, causing there to be neural networks that lead to distinct and different spatial progressions that spread superior laterally within the transcentral hinal and hippocampal regions to the primary areas of the neocortex. The synaptic dysfunction and cell death is shown by the progressive loss of cerebral metabolic rates by the uptake of glucose and the progressive brain atrophy. The decrease in synaptic numbers within the gyrus of the hippocampus correlates with the declining cognitive functions. Amyloid changes are detectable in the cerebrospinal fluid with amyloid imaging, and structural atrophy may be detectable with MRIs. With all of these preventable understandings on how to test for Alzheimer's or early onset Alzheimer's, we can figure out what exactly a person has and how we could try to alleviate the stress. By looking at a healthy and diseased neuron, we can see how Alzheimer's affects it. When we look at the soma, we can see a lot of beta amyloid plaque. Due to that, there's a considerably less amount of signaling. When we go to the neurofibrillary tangles within the dendrites, we can see how can signaling occur from that. And even further, when we see cortical atrophy on the axon, when no membrane resistance is there, then there is no signaling. To end this video, we're going to talk about how treatment is developed for early onset Alzheimer's disease. As we know, Alzheimer's disease has yet to find a cure, but that does not mean the efforts to try to alter and fix a bit of its mental impairments stops. So far we have figured out that through the use of medicinal usages, we can stop a little bit of containing and holding on to a bit of mental functioning. This includes donepezil, rivastigmine, galantamine, and memetin. These all are medicines that will allow a little bit of sensation that the person who has Alzheimer's disease is still there with the crowd. Not only this, but we have started doing many trials, clinical trials, for humans to find out exactly how we can stop and prevent more Alzheimer's deterioration. Specifically, the main treatment right now for mild and moderate Alzheimer's disease is with the cholinesterase inhibitor. Positive cognitive and global symptomatic effects compared with placebo have been reportedly mainly in 6-month studies of cholinesterase inhibitors. All of these is based on a touch and base go. We have yet to understand exactly what is the stoppage for Alzheimer's, but that does not mean we will stop in general. Alzheimer's disease is a crippling and deteriorating disease, but through knowledge and understanding, we will be able to diagnose, prevent, and treat.